Hello, my name is Monsi.
And I'm Lauren.
And today we're going to talk about protein synthesis. DNA, or deoxyribonucleic acid, is found in the nucleus of a cell and is responsible for building proteins. It is also called the blueprint of life.
All DNA is made up of subunits called nucleotides. Each nucleotide is made up of sugar, called deoxyribose, a phosphate, and a nitrogen base.
All DNA is a double helix shape, which is comprised of two strands of nucleotides that are twisted like a ladder. The nitrogenous bases on both strands are connected by a hydrogen bond.
These bases can only bond with a specific partner base. Adenine bonds with thymine, and cytosine bonds with guanine.
All of these components put together form a complete DNA strand.
This is what a model of DNA looks like. You can see here the double helix shape. Now we're going to talk about mRNA, or messenger RNA, which is another important component of protein synthesis.
Just like DNA, all mRNA is made up of a nucleotide subunit. These subunits consist of a sugar called ribose, a phosphate, and a nitrogen base.
mRNA is a single-stranded helix shape. Unlike DNA, it is only comprised of one strand of nucleotides. There are 4 specific bases that can be presented on the mRNA nucleotide. They include uracil, adenine, cytosine, and guanine. Here is a model of mRNA. You can see how it only has one strand.
Now that we have talked about mRNA and DNA, we can talk about protein synthesis. There are 2 steps to protein synthesis: transcription and translation.
Transcription occurs when the mRNA enters the nucleus and copies the DNA. During the copying process, the nitrogen base from the DNA are mapped onto the mRNA. Guanine maps to cytosine, thymine maps to adenine, adenine maps to uracil, and cytosine maps to guanine. It is important to remember that mRNA does not contain thymine.
The mRNA then leaves the nucleus and goes on to attach to a ribosome. This is important because DNA cannot leave the nucleus. As you can see, it is under cell arrest. This is important because mRNA is solely responsible for bringing the DNA data to the ribosome. At the ribosome, translation occurs.
Before we talked about translation, it is important to know that a codon is a grouping of 3 bases on our mRNA, and an anticodon is a grouping of 3 bases on tRNA. Each codon will have a specific complementary anticodon. Remember the base pairing rules mentioned before in this video.
Guanine pairs to cytosine, cytosine pairs to guanine, uracil pairs to adenine, and adenine pairs to uracil. Remember, neither tRNA or mRNA have thymine.
The mRNA will move from the nucleus through the cytoplasm and attach to the ribosome, where it will meet tRNA, or transferred RNA. It is important to know that the tRNA has brought a specific amino acid that corresponds to its anticodon.
As the codons and anticodons of the mRNA and tRNA connect, the specific amino acid will form peptide bonds between themselves. The type of amino acid tRNA brings with it is dependent on the anticodon of the tRNA. This is how the mRNA will be able to program the amino acids. For example, the anticodon containing the correct amino acid will connect to the correct codon on the mRNA.
Soon, we will be left with a polypeptide chain. When these polypeptide chains get folded together, we get a protein.
Well, I hope you can telomerase all your friends that you liked our video. The end.
We're very sorry we couldn't procure a protein pun for you, so you're just going to have to settle for one that involves telomerase.
Thank you.